Okay, so the first step of constructing a phylogenetic tree is you take the raw data, and then first of all you need to create a new alignment, and then you choose DNA, and then you can see there is a sequence, and then we click the sequence. First of all, we have to delete it, and then we try to find the insert sequence from file where we put the raw data, and it appears on the screen right now. And then we put align by cluster W options so that we can see the colors, and then we wait till it loads. So we have the colors according to the letters of the DNA. And then the next step is we check one by one whether there is an empty gap on the sequences, so that it won't alter or it won't change the results of our phylogenetic tree. And don't forget to align the DNA sequences so we can see the differences of the gene between all these species. We can align the DNA sequences by putting data and export alignment, and we are going to use the FASTA format, and then we put save. From the available dataset, we choose it, and then we repeat the same thing, but instead of using the DNA sequence, we put on we click on the nucleotide sequence. You can see all the based on the table, we can see at the top it is the best model that will be selected. It is automatically selected on the top. And here comes the next step, which is constructing the actual tree that we are looking for. You can see, just use all these settings based on the informations that we have previously. Okay, make sure that the model and the method is the correct one so that it won't change the results. And we have the phylogenetic tree, and what we have to do is save the file, and we are set to the next step, which is answering and interpreting the tree topology.
So as we can see from our phylogenetic tree, we can see that species Z and species 6 are more closely related to one another than either is to species 1. Species Z and species 6 share a shallower knot than knot that share with species 1. Taking a broader view, we know that species Z, species 6 and species 1 are more closely related to each other than to species 8 and species 15.
Based on the phylogenetic tree given, species Z has the most genetic changes. This is because, in most phylogenetic trees, branch lengths are proportional to amount of genetic change. Longer branches typically indicate more genetic change. As stated in Digital Atlas of Ancient Life 2017, branches on a tree are scaled so that they reflect the amount of evolutionary change that has occurred. In this type of diagram, branch lengths will differ, and taxa will not be aligned at the branch tips. Sometimes, this type of tree can be called a phylogram.
For question which taxa are closely related, the answer is taxa SP16, 9, 3, 4, and 7. SPZ and SP6 are second closely related taxa. SP16, 9, 3, 4, and 7 are the most closely related due to them sharing the same most recent common ancestor and having similarly length branch, indicating less genetic changes between them, while SPZ and SP6 also share the closest common ancestor. The branch length difference between SPZ and 6 shows significant genetic changes between the two taxa. For the next question, which taxa is the outgroup? And the answer is SP12. The reasoning behind it is because we can see from the phylogenetic tree, the SP12 has so many dissimilarity to the other groups, which is we consider as an in group.